Phospholipids are lipids made up of glycerol joined to two fatty acids in a phosphate group. The fatty acids made up of hydrocarbon chains are nonpolar, so they act as hydrophobic tails. On the other hand, the phosphate group has a negative electrical charge and acts as a polar hydrophilic head. When introduced to an aqueous solution, phospholipids arrange themselves into a bilayer in which the hydrophobic tails make up the interior and the hydrophilic heads the exterior, making it so that the hydrophobic heads are in contact with the aqueous solution. Both on the inside and outside of the bilayer, this structure, phospholipids, is what makes up the plasma membranes of cells. There are two different types of proteins in the plasma membrane, integral and peripheral. Integral proteins penetrate the hydrophobic interior, and the majority are transmembrane, which go through the entire membrane, but some only extend partially into the hydrophobic interior. These hydrophobic regions consist of one or more stretches of nonpolar amino acids, and hydrophilic parts are exposed to aqueous solutions on both sides of the membrane. Peripheral proteins are loosely bound to the surface of the membrane and expose parts of integral proteins. The name fluid mosaic sac model comes from the membrane's innate fluidity and the membrane proteins arranged in a random mosaic along the phospholipids. Plasma membrane fluidity results from the weak links between the phospholipids, keeping them apart until the temperature drops and they settle into a packed arrangement. Membranes composed of unsaturated phospholipids are more fluid because the kink in their tails prevent them from having a compact structure. The proteins embedded in the plasma membrane, which forms a mosaic-like structure, provides a strong framework for animal cells when they attach to extracellular matrix. The cholesterol, known as fluidity buffer, reduces membrane fluidity at moderate temperatures and prevents solidification of the membrane at low temperatures. Glycoproteins are carbohydrates attached to proteins, serving as identification tags that are recognized by specific membrane proteins of other cells. Glycolipids are carbohydrates covalently bonded to lipids, and they act as receptors and perform cellular contact and signal transduction. The selective permeability of a cell membrane is influenced by the phospholipid bilayer and transport proteins. Small nonpolar molecules can interact with the hydrophobic interior and pass freely through the membrane via diffusion without aid. Larger polar molecules are unable to pass freely and depend on transport proteins. Carrier proteins shuttle specific molecules across the membrane by changing its shape when it binds to a certain molecule. And channel proteins have a hydrophilic channel that acts as a tunnel for certain molecules and ions. For example, aquaporins, a channel protein, are specifically designed for water molecules which diffuse through the protein into a cell. In plant cells, cell walls provide structural support with turgor pressure in a hypotonic environment so that the plants stay upright. Passive transport is the movement of molecules down the concentration gradient without any energy. For example, diffusion is the movement of molecules to spread evenly into an available space, and osmosis is the diffusion of water molecules through a selectively permeable membrane. Facilitated diffusion is a passive transport aided by transport proteins down a concentration gradient, with channel proteins acting as a tunnel and gated channels opening or closing in response to stimulus. In active transport, molecules move against their concentration gradient or electrochemical gradient via carrier proteins and ATP energy. The sodium-potassium pump, or ATPase, is a type of transport protein that moves potassium-positive ions into a cell and sodium-positive ions out of a cell, maintaining a high positive potassium ion concentration in the cell and low sodium-positive ion concentration in the cytoplasm. This is facilitated by a subtle shape change in the protein triggered by ATP's phosphorylation that changes its affinity from potassium-positive ions to sodium-positive ions, and vice versa. Large molecules move in or out of the cells via exocytosis and endocytosis, with exocytosis sending substances out of the cell while endocytosis brings substances in. These bulk transport require direct use of ATP in order to engulf particles with the membrane. During exocytosis, transport vesicles bud from the Golgi apparatus and move towards the plasma membrane via microtubules. In endocytosis, areas in the plasma membrane form a pocket or vesicle containing extracellular material through three ways. Phagocytosis, also known as cellular eating, is when a cell extends pseudopodia outwards and engulfs the particle. Pinocytosis, also known as cellular drinking, sees an area on the membrane sinking inwards to take in extracellular droplet molecules in small vesicles. Lastly, receptor-mediated endocytosis is a specialized type of pinocytosis that human cells use to take in cholesterol, in which receptor proteins are embedded in the membrane and cluster in coated pits. When signaled, then the membrane sinks in and it brings the bound molecules as well as some other small droplets into the cell. Once particles have been liberated, empty receptors are recycled to the plasma membrane by the same vesicle.